so excited to introduce Dr. Snyder is the Stanford Ascherman Professor of Genetics and the Director of the Center of Genomics and Personalized Medicine. He received his Ph.D. training at the California Institute of Technology and carried out postdoctoral training at Stanford University. Dr. Snyder has pioneered the use of big data and multiomics to advance scientific discovery and transform healthcare. His laboratory has invented many technologies that are widely used in medicine and research, including methods for characterizing genomes and their products, for example, RNA-Seq, NGS paired end sequencing, Chip-Seq, and later Chip-Seq, protein arrays, machine learning for disease gene discovery. His application of omics and wearables technologies to perform longitudinal data profiling of people when they are healthy and ill is transforming medicine and healthcare. Indeed, his laboratory's recent work to use smartwatches and wearables to detect illness, including infectious diseases such as COVID, prior to symptom onset has been used by many thousands of people. He's also helped co-lead many large-scale projects, including ENCODE, HMP, HuBMAP, and HTAN. He has founded 16 biotechnologies companies, including Personalis, Qbio, January AI, Filtricine, and RTHM. Thank you so much, Dr. Snyder, for joining us today. We're so delighted to have you and hear about your work.
Great. Well, thanks for having me here. It's fun to be here. And I think we're small enough if you want to ask questions either in the chat as we go along. It'd be hard for me to see a Q&A if there's one open. But I am going to talk about our work on big data. Some of it is things that some of you may have seen me talk about before, but we do have some new updates, especially around microsampling, that I'm looking forward to presenting and hearing your feedback on. All right, so I guess I should start by saying I'm conflicted in absolutely everything I'm going to tell you, so you don't have to trust any of it if you don't want to. All right, so many may have heard me say this before, but I think healthcare as we currently practice is broken in many different ways. First of all, it's more like sick care, where we tend to treat people once they're ill, as opposed to keeping them healthy. But even if they are healthy and you go to a physician's office for a visit, the way it typically works is shown here. You know, you have to get in your car, travel sometimes, depending where you live, that can be a long distance. You arrive at an office, it pretty much looks the same way it did 40 years ago, maybe with a few new gizmos, but for the most part, it's pretty similar. They'll draw a very, very large aliquot of blood with a needle, which can sometimes hurt. From that large aliquot of blood, they'll make typically very few measurements for that and physiological ones. Some of them, I would argue, are even questionable value. And then once they get the data back, they make decisions on you based on population-based averages. So we think there are many aspects, if not all aspects of this, that are wrong or could be improved. And with regards to this last one on population averages, you've probably been told since you were little that your oral temperature when you put a thermometer in your mouth is 90.6. But if you actually look at the data that are out there, and this is a very typical study, it's more like this, 97.5. I personally am 97.3. I've been dropping a little bit the last 8 years. But the more important point here is that there's a spread. This is a 25th quartile, 94.6. This is a 75th quartile, 99.1. And what that means in today's world is that if you have a normal baseline temperature of 94.6 and you go to a physician's office and they measure 98.6, they'll tell you you're healthy, you're normal, everything's fine, you know, go home. But the reality is if you're up 4 degrees over your normal baseline, I guarantee you're not healthy. Something is off. And that's a big theme of our work. Try and figure out what your healthy baseline is so you can detect illness by seeing these shifts from that. So your health is influenced by many things. I think we all know this intuitively. Your genome, you know, activity, food, all these things, including stressors. But we're in a world where we can now quantify a lot of this, you know, certainly sequencing genomes, getting activities from smartwatches, food logging, still clunky, but doable, stress indirectly. And certainly we're doing a lot now in environmental exposures. But equally importantly, I would argue you can measure the effects of these things by actually looking, you know, looking at their impact on molecular and physiological profiles. And so a number of years ago, we set up what we call personal omics profiling. The idea is that we would do deep data dives on people. So basically, we would sequence their genome, that's just done once, But then we would make other measurements, mostly out of blood. So for epigenomes, methylome, transcriptome, proteome, these we do out peripheral blood monocyte cells, your immune cells. We also follow cytokines, metabolites, lipids out of your plasma. And we have 4 different microbiomes we study now: gut, nasal, tongue, and skin. That turns out to be quite interesting. This is on top of deep clinical tests, questionnaires, some advanced tests like stress echocardiograms, glucose control measurements. We'll get into that a little bit. And then also doing a lot with wearables, started this back when they were mostly just fitness trackers. And so one aspect is, as I say, it's a steep data dive. But the other aspect is doing this longitudinally. So we sample people every 3 months while they're healthy. And then if an adverse event comes along, like a viral infection, we take 5 to 7 additional samplings to see what's going on during that period. And we've been running this actually for a number of years now in a smallish group, 109 people. I'm one of them. We've been running it 13 years on me, and it's 10 years come March for the cohorts, and a little longer, some a little less. And just from the first 3.5 years of profiling, we actually had 49 major health discoveries, meaning half the people learned something pretty important about their health. I should say the reason we're doing all this is trying to understand what it means to be healthy. You know, how do people change over time? What happens early signs of illness? And relevant to this slide is, you know, can these advanced technologies like genome sequencing such be powerful for you know, managing people's health? And as I say, nearly half the people learn something pretty important about their health. And it spans a wide range of areas: hem-oncology, cardiovascular, metabolic. And you know, some of these were a big deal. We caught some with early lymphoma. These are 2 precancers. There were 2 people with serious heart issues from genetics, another from wearables. And so we actually caught all of these things pre-symptomatically, meaning they had not yet had symptoms, but by doing a deep data dive, we could see things were off. And then follow-ups actually verified or basically pinned this down. And no one technology did it. Sometimes it was a genome sequencing, sometimes a wearable, sometimes it was imaging or molecular. Usually it's combinations of markers would reveal these. And so we think of this as basically, if your health is a 1,000-piece jigsaw puzzle, the way we do it today is look at about 6 pieces and try and figure it out. Whereas we're trying to look at more like 600 or 800, something like that. Just a much, much clearer picture of your health. And to run a few examples, genome sequencing was quite powerful. So from the first 70 people, I need to update this, but 12 from Mendelian learned something pretty important about their health. And for 3 of these, it was a big deal. Like this individual actually had a mutation in a cardiomyopathy gene, follow-up tests with stress echo. Sure enough, his father died of a heart attack, and he did a stress echo follow. And sure enough, he had a heart defect. He's a young guy, he's actually on drugs now. One individual there were 2, but one individual who had a mutation in this gene did a whole body MRI follow-up, and sure enough, they actually had early thyroid cancer, which was removed. They were able to keep most of their thyroid, and that was good. And then, one individual had a BRCA mutation. So far, all fine there. 9 individuals were thought to be diabetic coming into the study. One of them was actually, they're all thought to be type 2, but one of the individuals turns out has a mutation in this gene. They're not really type 2 at all. They're really a MODY mutation, which is a different kind of diabetes, and they've been on suboptimal medication for years. So I apologize. There's some sort of talking going on outside. I hope it's not showing up there, but I'm going to shut an extra door and ask them to be quiet. I'll be right back. I apologize.
Yeah.
All right. Sorry about that. All right. Well, like I say, it's kind of informal, I suppose. Anyway, here is, so these are the Mendelian cases, again, where we learned something pretty important about people's health. And even the BRCA person will get screened more frequently as a consequence. I think most of you know, certainly geneticists know, that these Mendelian cases are certainly nice and solvable and such, and good for risk predictions. But most diseases are really complex disease. They're really, due to, they're thought to be due to lots of variants of low effect. And so single genes can actually kill you, if you will, by a single mutation, but these cases are thought to be, again, these common variants of low effects. And I like to think of this as, killing you by a sledgehammer versus death by 1,000 cuts. And the way people deal with this now is something called polygenic risk score. As many of you probably know this, but these days people will sum over, thousands, if not millions, of single nucleotide polymorphisms or single nucleotide variants, common ones, and make predictions about score. And it does work for the top 2 to 3% if you're at the very, very high end of the risk. And there's some nice plots to show this, but it really doesn't work for most people. And the reasons for that. And we think some of the main reasons are this is done by linearity. Basically, you're summing these up in a linear fashion. And anyone who does genetics knows genetics isn't linear. 1 + 1 is rarely 2. If you have 2 mutations in the same pathway, 1 + 1 is 1. If you have 2 mutations, separate pathways, 1 + 1 can be 15. So we're basically, we think that's a flaw. It also ignores rare variants of the sort I just described. So we've come up with new ways of analyzing genomes. This is a bioinformatics symposium. We've been using machine learning, which is obviously popular for lots of things these days. We started this kind of early on to see if it would be valuable. And we're using it in 2 ways. Right now, we're working on what I think is the optimal way, but I think these 2 ways have got us on the right track. The first one is this case where we actually wanted to look at rare variants, which, as I said, have been ignored from the complex disease standpoint. And what we've done here is we took on this disease. It's called abdominal aortic aneurysm. It's known to be highly heritable, 70%. And we thought it was a good one because the way it's discovered is that people's aorta bursts and they die. So we think that's a terrible way. We need to know who's at risk. And so we thought this would be a very, very good one to take on. And Phil Sal, who is at Stanford, close collaborator on this, basically had a great cohort. So what we did was something that I think freaked out a lot of GWAS folks. We just sequenced around 401, 268 cases, 133 controls. We did a whole genome sequencing, but we mostly zoomed in on the coding regions where we called pathogenic mutations. And then sprinkling, there's a limited amount of EHR data. We do machine learning and say, give us all the features of genes, the ones with pathogenic mutations that are enriched in cases versus controls. And I should point out there were 3 genes implicated, very, very sketchy. But none of them well accepted. And when we were done, we came up with 60 genes running this, did tenfold cross-validation. Wound up finding 60 genes, great p-value, 10 to the minus 13. Tenfold cross-validation, I may have said that already. And then when we looked at them, it turns out, they're exactly in the kinds of things you would expect. Blood circulation, blood pressure, there's a protein interaction network, cardiomyopathy, this is not all, it's a subset of genes in here. And same with aneurysm. And we went on to show the genes are misexpressed in patients with AA, and mouse models actually have aneurysm kinds of defects, and they also are misexpressed. And so we could even go on and build a risk score, if you will, where the genome alone, it's not bad, it's 0.7. You might say EHR is good. That's true as you get later in life, but together they're even more predictive. I know that doesn't seem like a big boost, but it actually helps a lot in the specificity. So at the end, it's a decent risk score, if you will. It's as good as what they're using for high risk for cardiovascular disease. And so we think, it has some value. So we're trying to roll this into the clinic. So this is using rare variants and combinations of rare variants that we think would contribute to this disease. Well, what about the common variants? We decided to try also to take that on. Here's our second approach. We're using this we used it and published it. We're using it for a lot of things now, but we published for ALS and COVID severity to try and get at the genetic basis of each of these cases. And I'll go through them. So for ALS, you probably know it is a genetic disease, something like 61% heritability. And 7 genes have been identified when we started the study using GWAS. And so here what we did, we used a somewhat different approach. We started with mapping the open chromatin regions of motor neurons, which is certainly one of the major set of neurons that are involved in ALS. And so we mapped the open chromatin regions. And then what we did was we took the GWAS signals, all the GWAS signals, not just the significant ones, all the GWAS signals. And we did machine learning here saying, give us all the open chromatin regions that are enriched in cases versus controls. And there was a giant GWAS study of well, you see it here, 12,000 cases, 23,000 controls. And the net result was it worked quite well. We basically could pin down, we went from 7 genes to 690 genes, 100-fold more, because that's a big GWAS study. We could validate with another GWAS study. And we went from 6% of the known heritability to 36%. So we picked up many of the well-known ALS genes, and we showed that this list is significantly enriched for everything implicated in ALS, meaning misexpressed genes, et cetera, as well. We even went on to show that these genes are downregulated both in brain tissues of ALS patients and also in iPSC motor neurons. So the genes are generally misexpressed, lower expressed, in fact, it turns out. So we think it's lower function. We then could go on. And so that was a big deal. It did show a list, and a lot of people are actually using this list right now of genes. And we could go on, same thing with protein interaction maps, and see where they map in the interaction network and what kinds of processes. And it turns out a lot of the genes involved are in distal regions of the motor neuron. So we think it's not just the motor neuron, but the distal region in particular is particularly important for this ALS condition. And then we went on and took this even one step further, took one of our top genes called CANK1, and there were some, actually we'd seen in that earlier kind of analysis, there were some mutations in CANK1 that one might predict could actually be, ALS, could be responsible for ALS, important for ALS function. So we went in and made actually iPS cells and made that particular mutation from these what we thought were more closer to monogenic mutations in this, and then derived motor neurons, and showed they had all kinds of defects. They reduced survival, abnormal function, it's kind of shown over here. And even this hallmark of TDP43, this is thought, a lot of people think this is the cause of ALS. We could show that mutations in this led to mislocalization of this, meaning that the genes lie upstream of TDP43. So again, it suggests that TDP43, it doesn't mean it can't be causative, but they're upstream factors that are potentially causative of this as well, which is kind of an important concept for this field. So that was wonderful. As I say, we found 10 to 100 times more genes. We went on and took this one step further for COVID severity. These things are always tough to get past reviewers, but we thought this was pretty cool. We did the same thing, but now we did it for COVID severity where the cell types were not so clear what was responsible for the disease. If you look at the literature, some people say, well, it's endothelial cells, NK cells, T cells, whatever. Every cell type's been implicated in COVID severity. I should say this is advanced age COVID severity. When people get old, their immune system is not as robust. They get COVID, they get hospitalized or die, or both. And this is a case where, a lot of young people get severe COVID and wind up in the hospital. And you say, what's going on there? Well, genetics is thought to be a big part of that. And so a giant GWAS study had come out that had about 5,000 cases, 1.4 million controls. And there was, basically the study had come out. These are cases that were 6 years or younger, so they're not the older group. And we basically did the same sort of thing, but here we use single cell data. We took lung data where there are 19 cell types, including all your immune types, NK, other cells, macrophages, what have you, along with epithelial, endothelial, what have you. And we did the same kind of concept. So we had open chromatin regions mapped out for multiome analysis, it's called. And we could, same thing for each cell type, take the GWAS signals and ask, are any of them enriched in cases versus controls? And what regions are there? And in the end, we came up with 6,600 regions, which mapped to about 1,400 genes across these 19 different cell types. And some were found in multiple cell types, also an exaggeration, about 3 or more means orange. Some were unique for very specific cell types. These are just the 19 cell types here. And we went on to show that if you look at where the signal's most enriched, it turns out it's in NK and NT cells. Both have very, very strong enrichments for these genes and the genetic signal, if you will. And at the end of the day, we could basically say for the most severe COVID cases, we could capture 77% of the heritability, which is kind of unheard of. I think it's because it's such a large GWAS study. And so again, so we could find this out. I may have forgotten to say there were 47 genes known before, and we boosted this up once again. Not quite 100-fold, but many, many-fold. We've now done this for many diseases, working very, very well. So the bottom line, we think this machine learning works for 2 reasons. One is, first of all, restricted the space to open chromatin, that helps. But the others, we're using machine learning, which is not linear and therefore much, much more powerful in these cases where we think, genetics is great. I'm the chair of genetics. I like genetics, but it's not linear. And I think that's where machine learning has a huge advantage. So I'll tell you, genetics is wonderful. Oh, we don't have predictive models yet. We're working hard on this. I'm pretty sure we can get there. But we have predictive models for prognosis of ALS, but not for who's going to get it. We're working on that. All right. Other technologies we think are important for finding disease. This is the case of the spleen, the early lymphoma. Imaging of spleen happened to reveal it was enlarged and there were some blood markers that were elevated. So that's how that person's disease got detected. There's a young guy who actually had way too much IgM, it's a particular protein antibody, some of you may know. His 2 samples had way, way more than everybody else. There's 1,000 samples here. And as I say, and then they did follow-up experiments. Sure enough, he's got this pre-cancer that can convert to aggressive cancer. So it's kind of useful. We saw it, he'll get monitored more frequently. We're very interested in type 2 diabetes. Some of you may know I'm type 2 diabetic, and it was actually predicted from my genome from a polygenic risk score. It worked for me because I'm at the high end of the distribution. But coming in the study, 9 individuals were thought to be diabetic, but it turns out 2 were diabetic who didn't know it. And many, many were pre-diabetic who didn't know it. And that's very common. 9 out of 10 pre-diabetics don't know it. And 70% of them will become diabetic. So we could reveal that right away. And we're very interested in how do people become diabetic, because it's not so clear, but because we're tracking people so closely, we can see a little better what's going on. And what we discovered is that 9 other individuals became clinically diagnosed as diabetic, and others had diabetic range values. So this is just in the first 4 years of the study, and others became pre-diabetic. And we expect this with aging. I forgot to say the cohort as a whole was 53.4, a bit older, which is probably why we're making lots of health discoveries. So of the 9 who became diabetic, how did they get there? Well, 7 got there by gradually seeing either their fasting glucose, see in orange, or something called hemoglobin A1C. It's a marker for glycosylation levels, and it gradually went up. And then this person intervened. So this is the common scenario. 7 of the 9 people gradually increased in their fasting glucose or hemoglobin A1C, or even something called oral glucose tolerance. It's a different kind of test, but it measures your ability to consume glucose, if you will. So they gradually got there. And 2 of them had gained weight, so they hit the canonical, how you get type 2 diabetes, you gain weight and you have problems. The other 5 did not gain weight, so they just gradually got there through aging, if you will. 2 individuals got there through as though something triggered it. This one's me. I have 8 years of data. I first became diabetic. My genome predicted it. But I first became diabetic after a very nasty viral infection, it turns out. It's a respiratory syncytial virus, which is flying around right now. So basically, I actually was going in for an insulin resistance test. Turns out I was sensitive, but we spotted that my fasting glucose was high. We were both surprised. Went in a week later and got measured, and sure enough, crossed the threshold to be classified as diabetic. And it turns out, it's arbitrary. 6'5" is called diabetic. So in my case, I was able to do a lifestyle intervention. I doubled my biking, started running. I cut out all sugars. I had a bad diet before all this because I didn't know I was at risk. And so I basically cut all that out, and I brought it down to normal levels. That's several years. And by the way, this is not a tiny blip. That's about a year of data. And so I got it down to normal levels. In fact, it was so boringly low, I stopped looking at it. And then someone else spotted it here. I said, Mike, do you realize your hemoglobin A1C is 7.0? And I said, I had no idea. I got up to 7.1. And I had stopped running, guess when? Right here. And I also got a second viral infection. I actually had several, but this is certainly coincided. So it's probably a combination of both the infection and the running, because I started running. I'd stopped running. I started running again. I could get it down, never way down to the original normal level, but down somewhat. And bottom line is I basically kept running then. I was running about 4 days a week, 4 to 5 miles a run, 45 minutes or whatever. And it gradually kept going up in spite of all my running. And so I had heard muscle mass is better for glucose homeostasis. So I switched from running to lifting weights. You can't tell, but I did gain 10 pounds of muscle mass. I do whole body MRI too. So we could see that. That didn't work. I just kept going up anyway. So finally, as I was getting into I was crossed back in the diabetic range. As I was getting towards the high sixes, I went on metformin, and that failed miserably. That's a frontline drug for type 2 diabetes. Went all the way up to 7.5. I kept increasing the dose. So what's the solution for me? Oops, looks like I forgot the slide there, but basically it turns out more data. So I am insulin sensitive. I make insulin fine. I just don't release it from the pancreas, which is called glucose disposition. We can see that. And there's a drug for that. It's called rapinolide. And that actually works pretty well for me. These days I'm on SGLT2 inhibitors. They work quite well, but also the thing that's working the best is this new miracle drug you may have heard about, GLP-1 agonist. So I'm on one of those, and that works incredibly well. So we call this precision diabetes. By better knowing what's wrong with me, I could take the right medication. As I said, metformin didn't work. And so we've been pinning that down. It's a question in chat. Oops, I see, that's the streaming thing. Never mind. Okay. All right, so that's my diabetes story. And again, we could actually run these tests. The ones, everything we're doing costs millions of dollars because it's a research study. But you could do all this precision diabetes actually relatively inexpensively, we think, and get people on the proper medication right from the get-go. All right, one of the things we learned as we were profiling people is they have stable profiles that are really quite stable over time, meaning your metabolites, what have you, and they're quite personal. So the difference between you and someone else is quite large compared to the difference between a disease you, say a viral infected you, and a healthy you. So we think these personal profiles are really, really important to capture from a health monitoring perspective. And you'll see more of that in a minute. But the other thing we could learn, because people's profiles were generally stable over time, we could see, well, maybe some markers do change. We do. So we had some people whose markers change. Well, not everybody's markers change seasonally, but there's seasonal patterns in the data, which was pretty cool. In California, we say there are 2 seasons, it turns out, based on the patterns. But we could also see how people age, because we're following what, their markers over time. And we think you need as few as 5 measurements within 2 years, and we can tell you how you're aging. And so we had 43 people with enough data at that time. We have more now. But this person is me. I'm a typical ager. My coagulation, my metabolic pathway, they all pretty much go up. I have a lot more time points because I sample myself a lot. This person, though, if you look at their top pathway, they're a cardiac ager. Their cardiac hypertrophic signaling pathway is the top pathway that's changing over time. And we later learned this person's stage 2 hypertensive, which sort of fits with this idea. Okay. And we went on and took all the people. We said, well, how many patterns are in the data? And there are 4. Now I know, and we call them ageotypes. Now I know there are more than 4 ageotypes, like the cardio one, but we're not statistically empowered to find them all. But there were 4 that we could find that were statistically significant. So each row here is a ageotype. Here they are: kidney, liver, metabolic, and immune. And then each column is a person. And so the people over here, they're aging in all 4 categories. This person's aging in, 3 of the 4, not so much a kidney ager. I'm in the middle. I'm not much of an immune ager, which I think is good. This might be me, but I'm more of an ager in kidney, liver, and metabolic. This person's kidney ager, kidney, metabolic, and so on. So we can see how people are aging. And not only that, there are clinical markers associated with this. Oops, I left that out. With each of these ageotypes, so you can actually act on those. So the obvious one is if you're a metabolic, turns out hemoglobin A1C tracks with this. And so we actually have people who are reversing their hemoglobin A1C because they've done lifestyle changes. Again, that's a case where you could actually do intervention. So we think this is valuable information that you might, modify your behavior. If you're kidney ager, maybe you should drink more water. If you're liver aging, maybe you shouldn't do so much binge drinking, and so on and so forth. So we think the information's actionable. One question I get a lot is, how are you going to scale this? How are you going to get out to millions of people? It costs a fortune. And the answer is, well, I'm in Silicon Valley, so you form a company. So I'm totally conflicted here. But the point out of this is that we've done a medical version of this, much, much deeper data dive. And it also adds on whole body MRI, which we think is quite powerful. And so it turns out that, just from the first 100+ people, we caught someone early in ovarian cancer, prostate, even someone with pancreatic cancer, which is almost never discovered early. Again, all pre-symptomatically because we're doing these deep data dive, and so on and so forth. So we can find these things in advance of symptoms through this deeper data profiling people. And so it's not cheap. It's $3,500 per visit. But, if you get a heart attack, that's super expensive. So we can discuss what's relevant. I know we can get the price down long enough. So anyway, that's one. We think this data profiling is going to be super powerful. Now, the other thing we've been working a lot over the last few years, and I should be able to save time for questions here, plenty of time, is this issue about going to a doctor for a visit, which it's suboptimal for many, many reasons. One of which is when you go there, everybody's sick, or a lot of people are sick. So you don't really want to be healthy and go to a doctor and get sick, kind of counterproductive. So we think remote monitoring can be powerful. We're doing this in 2 ways. One is Wearables, many of you may know this, and the other is microsampling, at least for sampling remotely. The analysis is done offline. So Wearables, you probably know, there's now there's actually over 1,000 different devices out there, many in the smartwatch, but other arenas as well, including rings and such. And so, and I'm keen on this. I wear a number. If you look up, I'm wearing 4 smartwatches right now. None of a ring, but I got my CGM, my continuous glucose monitor. We'll talk about that too. Many different devices, including my exposometer. And so one of the things we discovered early on, I want to kind of get into the thick of this, is that we could pick up infectious disease from a simple smartwatch and a pulse ox. And it was the way we got into this is when I discovered my Lyme disease, of all things, using this pulse ox and smartwatch. And the story was, I was helping my brother put up fences in rural Massachusetts, and then 2 weeks later was flying to Norway. And you may or may not realize, but your blood oxygen drops on airplanes because they don't pressurize the cabin fully to atmospheric pressure. And so it would normally drop. And on this plane, it dropped when I was flying to Norway, way, way lower than normal. I was on Frankfurt to Norway. And it was a median of 90 for my blood oxygen, never came back to normal after landing. And so I also saw my temperature was up for my smartwatch. It's kind of low resolution. I later learned my skin temperature went up as well from the smartwatch. And I then got a fever off and on. I did go to a physician there in Norway who drew blood, so my monocytes were up, and he basically wanted to give me penicillin. And I said, no, I need doxycycline. Got a little tense there. But he gave it to me. You take it 2 weeks. So when I got back, sure enough, I was Lyme positive. I got tested. I even had some antigens there. And I'd given blood, well-controlled experiment 3 days before I left, and I was negative. So I seroconverted during this time. So, so that was quite powerful. That this is all pre-symptomatically I detected Lyme pre-symptomatically from the elevated heart rate and blood oxygen. So, of course, I won't show you this, but then we showed that we could pick up respiratory viral, including asymptomatic ones, retrospectively with the smartwatch data. And I won't get into that, but we published this a little bit ago, excuse me, 2017. Excuse me. And then, of course, along came COVID. We've been improving the algorithms, this infection detection work. And then, and also building an infrastructure to be able to scale this. And this is just the first wave. So this is May of last year. Excuse me. I think these days we're around 45,000 cases a day, which is way underestimated. Nobody bothers reporting it anymore. And so the point is that here it is. It's here to stay. And, can we apply some of these methods to COVID and other things? And so the answer is yes. We spent a lot of time on it. As soon as the pandemic hit, we very quickly started a study of 5,300 people, 32 of which we partnered with Fitbit, 32 of which were wearing a Fitbit at the time they got ill. And this is our very first case. So this is an individual who basically, sorry, got a little frog in my throat there. This individual basically had a diagnosis day here. Their symptoms first appeared the day before. If you look at their resting heart rate, it basically jumped up 9.5 days prior to symptom onset. So very, very clear signal. You can't miss that. I'll show you in a minute. We have a real-time detection method. You get somebody's baseline, you look for a shift up, an anomaly signal on top of that, certain number of standard deviations, magnitude and time. And and it worked. It worked in 26 of 32, 80% of the cases. And it was a median of 4 days prior to symptom onset. We would see and this works well for COVID because it's a long pre-symptomatic period. So we went on to build I want you all to sign up for my study, innovations.stanford.edu/wearables. You can write to me and I'll send you the address, or I'll send it to the seminar person who can send it out. We went on to build this real-time alerting system. So what it's doing is tracking your baseline. We had to come up with a new lighter weight algorithm, so we didn't break the bank on the computation side. But basically, it's tracking your resting heart rate and other parameters. Most of the signals around resting heart rate. We want to get more data in. It's harder to get than you think from the companies. But we built this algorithm. It works pretty well, actually. So this is, again, one of our first cases that you get a red alert if you've had a certain number of if you will, standard deviations of magnitude and duration of time above your baseline. So it takes your personal baseline, looks for the shift up, but you only need 2 beats per minute elevation to pick up the signal. And it does work. It works same number 80% of the time, including asymptomatic cases. We're calling Apple and Fitbit. We can show this. And here's one of our first cases. This is when this person got symptoms, but you can see they were getting red alerts for 3 days prior to symptom onset, and they were diagnosed the next day. And so it works, as I mentioned, in both for Apple and Fitbit. It works in asymptomatic cases as well as pre-symptomatic. Here's an asymptomatic case. There is a diagnosis day. Obviously, there's no symptom day. It's asymptomatic. And for 2 weeks, this person was getting red alerts. This is a big deal. It's not specific for COVID. So other things trigger the red alerts. I know if we get more data, we can get better at distinguishing this, more types of data in particular. Workplace stress is the #1 trigger of red alerts. We set the sensitivity, which through surveys, people thought was pretty good, so that the red alerts go off every 6 weeks. That gives reasonable sensitivity and doesn't overly burden people. Intense exercise will do it, meaning running a marathon, not just your usual jog, but running a marathon, your heart rate stays up for a few days, travel, other things. There's quite a few things that trigger this. Other infections too. We even picked up a mycoplasma infection. So it's more of a stressor detection system. As I say, I know we can probably subtype the stressors as we get more data. And I can tell you it worked for me personally. So when I was getting ready to go to a meeting in New York City, that would be here. So first of all, normally I have green alerts, this kind of raw data right off my smartphone. I had a particular family stress event I had to travel for, so some red alerts that day. But when I was getting ready to go to New York City, where you guys are, I woke up that morning and I had a little bit of congestion. Is it allergies? Is it what's going on here? Asthma? I don't have asthma. Allergies, or is it an infection? So I did a COVID antigen test and it was negative. I looked at my smartwatch and it was positive. I had a red alert. So what did I do? I got on the plane anyway, even though, because it was a negative test, even though I had a red alert. And then I show up in New York, and the next day I'm supposed to go to the meeting, but you have to do a COVID test to go. This is a real date, April 10th. I don't care if you know it. And sure enough, I'm bright positive. So I had to spend a whole week in a tiny little hotel room in New York, all because I listened to my antigen tests and not my smartwatch. And so we think, it's more sensitive than an antigen test, less specific. I didn't know it was COVID, but I don't do this again if I get get red alerts and I don't have an explanation like drinking too much or overly exercising or something, I won't get on the plane. I can tell you that. All right. So anyway, it also picks up long COVID symptoms. So people who have symptoms after long COVID will have an elevated resting heart rate. So we think it's going to be powerful for health monitoring for lots of different conditions. And I can come back to that in questions if you like. The other area we're pushing hard on is glucose monitoring. We're very keen on this because of my type 2 diabetes. We started putting these on normal and pre-diabetics. And what we discovered is that, some people do have good glucose control. Some are moderate spikers after meals, and some are severe spikers. And there are healthy people running, so-called healthy, based on their fasting glucose. And they're just as bad spikers as diabetics. This person's one of them. And so we classified people into glucotypes. So based on their level of glucose and spiking. And so here's low, medium, and high glucotypes, severe glucotypes. And so, and one of the things we discovered early on, as did Aaron Siegel at around the same time, is that different people spike to different foods. And this is Aaron's work. I like to show this because of this particular panel here. But basically, some people will spike to bananas, others bread, others pasta. It really depends on you. It's very, very personalized. And here's an example of this. Here's a person who spikes to banana, but doesn't spike to a cookie. Now, that's probably the only person on the planet that doesn't spike to a cookie. But this is more typical. This person spikes a cookie, but not a banana, and so on. So you get the idea. People will spike to different foods. One of the things we found is that, people will spike, some people will spike to peanut butter and bread, others have protein bars, which have carbs in them. Nearly everybody spikes to cornflakes and milk. That stuff's evil, probably should be banned. Could be worse than smoking, I'm not sure. But the point out of all this is that, different people spike to foods and some foods are bad. So we've gone, so we're doing a lot with this, and the CGM stuff, just wearing a glucose monitor can be very, very impactful. And so we, it's been off the company on this one too. And so, the other area that we've taken on, we just published this about 3 weeks ago, maybe a little less, is this idea of microsampling. So this is going to sound familiar to many of you, but I can tell you it does work. We spent a lot of time, we spent 6 years on this, trying to figure out the best methods for microsampling. We settled on two. This is the one that we published because it came out first, but choosing Neoterix Meacher devices, and now we also use Tasso devices, where they collect very, very small amounts, 10 or 20 microliters, depends on the size used. Then you FedEx it, and we'll process the samples in the lab, and we'll do these deep omics profiles, lipidomics, metabolomics, proteomics, and targeted assays for cool key markers. And so it does work. This is just, I can't show you all the controls we've done, but we looked at sample duration, sample temperatures, to see if they would have any effect on stability and interaction terms between the two. So you can see we put stored samples either right away or at 3 different temperatures. held them for different lengths of time. It's a big experiment. We did it for 2 different people. And then assayed the stability of proteins like cytokines, metabolites, and lipids. And what we found is that nearly all proteins are pretty stable in this format for measurement. Metabolites, generally pretty stable too. This is duration, this is temperature, and this is an interaction term. But you don't have to, by the way, go out for 5 days. So you can actually get around some of this. The lipids are less stable. It really depends on the lipids. So we know which ones are stable and which ones aren't. We just work with the stable ones, and we can, you can probably put corrections onto the ones that aren't stable. We haven't done that yet, but that's certainly doable. We did show that whole blood and plasma are pretty similar. Then we did 2 really fun studies I think you guys might like to hear about with this. So that's what I'm going to tell you. We did one study where we had 32 people. We had them drink an Ensure shake, this thing you can buy in CVS. So before we sampled them before, and then 4 different time points up to 4 hours after. And of course, not surprised, there's a lot of stuff in the shake. And so you see big changes in people's metabolic profiles. Hundreds of molecules change. And we can group them into patterns, like the metabolites come up first, lipids next. In general, cytokines depends on the person. And so, we can even pick up a lot of important glucose control molecules like insulin, C peptides related to insulin, GIPs and incretin, and PPs involved in appetite suppression. So all these things are a big deal for glucose control. I was a little bit surprised to see leptin went down after drinking the shake. I think we have an explanation. And overall, it turns out the interferons went down. I'll come to that on this slide. One of the key things we discovered is that everybody's different, very, very different. They drink the same shake, and usually they behave the same for the class of molecules. So these are carbohydrates. I'm showing 3 of them: fructose, lactic acid, pyruvic acid, and it could be amino acids, be a different class of molecules. And you'll see what's going on here. Like this person, every box is a different person. This person's carbs plummet after they drink that shake. This person's carbs skyrocket after drinking the shake. Here's another skyrocket. Several of these are that way. Here's another person whose carbs go down a bit. There's some others like that too in here, this person. So everybody's different to these different classes of molecules and how they handle it. And I think we grouped them into 5 classes. It turned out 28 people. 32 did it, 28 we didn't get, 4 people didn't get all the data from. All right, so what's really cool though is that I'll just use this one, inflammatory response. Some people had their inflammatory response suppressed when they drank the shake, and other people had an increased inflammatory response, which again, so we're all responding differently to these different foods, to the same food, I should say. And in some cases, this is a big deal. So pro-inflammatory is probably not so good. Anti-inflammatory is probably good for you. So it's so cool that people are reacting differently to this different foods, and that we can actually measure this. So we can measure directly. Here's a person whose carbs plummeted, by the way. That's the green one. I forgot to say the gray is the average. So if you're inside that, you're below average. So this person's carbs plummeted. Yeah. All right. So that was kind of fun. Then the other thing we did was we wanted to see, well, can you do dense monitoring of people? And so we did this on me. I basically took 98 samples, 1 every hour with some exceptions, for over 7 days. And basically, monitored myself in incredible detail. At the same time, wearing this glucose monitor, continuous glucose monitor, and a smartwatch, and doing logging of sleep and food. And what we discovered is that, not surprised, hundreds and hundreds of molecules. We're following 2,200 analytes, including, once again, some pretty key ones. And so we'll start with a very simple correlation, if you will. Caffeine negatively correlates with sleep. You say, oh, big deal. Everybody knows that. And that's true. But what was interesting for me is that I actually stopped drinking coffee by noon, which I thought was enough time to clear it, but I still had this negative correlation. So what I do now is I drink less coffee, and I try and stop even before noon, like 11 a.m. or earlier. And so to see, and I think it's improving my sleep, actually. So you do learn stuff that is useful, even if you think you follow yourself pretty closely. Now, the thing that got us the most excited is, can we correlate physiology and biochemistry and vice versa, biochemistry and other biochemistry. So what kinds of events precede what other kinds of events? And some of these are likely to be causal then. And a simple example of this I put down here just to illustrate the point. So we do this time-lagged correlation analysis to see if we can see what early events are correlated with later events. And here is a simple example, steps versus heart rate. No surprise. So obviously, if you take steps, your heart rate goes up, and we get that correlation. You can see right here. And it turns out, but the nice thing is you can quantify it. It's about a 1 minute lag between steps and elevation of heart rate. So we can do this for everything. And then at the end of the day, we make literally thousands of associations between biochemical markers, heart rate, CGM, and steps. And so green are metabolites, this purplish red here is cytokines and proteins, and this one in here are lipids. And so again, we can make thousands. Some of these turn out to be pretty fun and pretty cool. So let me start again with another simple one. This is CGM and your insulin C-peptide, insulin C-peptide. And what we discovered for me is that my C-peptide gets induced about 10 minutes post glucose elevating. So there's, 10-minute lag correlation, if you will. And that makes sense. Glucose should stimulate this insulin peptide production. It doesn't measure release, but it measures production. Now, here's another cool one. Here's a marker involved in Parkinson's. And this thing goes up, it turns out, 55 minutes prior to glucose. Now, this relationship had been suggested before. We didn't discover it, but we can quantify when it is for me personally. And if you think about it, you could actually then try to take things that might mitigate that maybe don't do that. And alpha-synuclein is thought to be induced by stress and other things. Other, unknowns, here's a good one where your glucose levels go up before this immune ligand. This thing stimulates your immune response. We think, and glucose is known to stimulate, your inflammatory response. And we think maybe this ligand is one of the events responsible, and that goes up about 15 minutes after glucose. So again, we can make these, and there's other things that are brand new. These we couldn't separate, CGM and TNF beta, which had never been described before. So they get a lot of things like that in the data. What we've done now is we built this dashboard, and we have this cloud-based computing system, I think I mentioned before, that you can display. It pulls in your wearable, your clinical, your omics data, and you can display things on daily, weekly, monthly pattern, whatever you need to visualize your stuff. And the ultimate goal, of course, is just to use data to better manage people's health. So I'm a believer people will be getting their genome sequenced before they're born, at least in the Mike Snyder world. And then together with biochemical and wearable data, we'll be able to better monitor people, predict disease risk, catch disease early, and try and have people live, longer, healthier lives. And so I'm fortunate I have an amazing team. I won't have time to go through these. I have a great clinical collaborator with Tracy McLaughlin, which I didn't put up here, but a pretty big wearables team. It's kind of the older team now. And then here's our microsampling group led by Xiao Tao and Ryan, have driven a lot of this. And sorry, this is an accident. This is because I was giving a talk for an applied thing, but if you want to do the microsampling, you can get a metabolic test through YOLO, or if you have long COVID, you can go with that one, I suppose. Anyway, that's the story. I think I left some minutes for questions, and so I'm going to stop sharing now and open up for questions. So thanks again. I see a few. Should I call them?
Yeah, feel free to take those questions if you'd like to, or I can, whatever you prefer.
Okay. Andre, do you want to go first? I'll just go left to right on my screen as the hands are up. So we'll take it from there.
Thank you. Thank you very much for a very interesting talk. So I have a question about increasing the specificity for detecting physiological signals during using the wearables, sort of for distinguishing between COVID and stress. Where do you think the future improvements will mostly lie? Is it in the more hardware side of the wearables, sort of picking up better signals or picking up some new signals? Or is it more on the software side of analyzing the signals that were given using some algorithm or a model?
Yeah, great question. It's both. And I can tell you right away, we should be able to tell workplace stress from a COVID infection. And just by pulling in respiration data, the hardest part is getting access to the data. And companies, and especially at high resolution, it's very hard to get raw data. Like Apple's the worst. Probably going to get myself in trouble here, but we just can't get raw data from Apple. And they're very hard to get any data. You can get some low, you get whatever you can get shared through HealthKit. Fitbit does a bit better sharing. Same thing, we can get heart rate variability, which by the way is a great health measure. We don't have any of that in because none of that was really set up when we launched this. We're pulling it in now, but it's not at the frequency I'd like to get it. So it's one of the reasons we formed our own company to actually be able to get very high resolution data directly and raw data, and can come right to us without going to the company. So, so anyway, we do that now. So I'm confident that, imagine just pulling your respiration data, that's going to tell you the difference between a respiratory viral infection and a workplace stress event. So I'm confident now. Will we ever get to the telling the difference between COVID and, influenza? I'm not so sure. We'll have to see, but we'll pull in the data and see what we can learn, and let's see if it can do that. And I think we had a paper on machine learning in this area that I hope will distinguish us. We need a lot more data. It's one of the reasons why I'd love to have all of you sign up. And so if you are wearing a smartwatch, please do. Okay, great.